My name's Craig Nitschke. I'm Associate Professor at the University of Melbourne, and I'm presenting this work along with my colleague Linda Parker, who's a postdoctoral research associate with me and has been working on this project. So this project is Project E5, as you can see, and it's around modeling the risk of key revegetation species that Melbourne water uses to a changing climate. So this project is really about trying to understand the role that a changing climate will have on the resistance, resilience, or vulnerability, choose your key term there, on key vegetation species that are used in re-veg work in the Melbourne water area. So what we're interested in is how are these species likely to respond, or how they can be influenced by a changing climate? Which of these species are at the greatest risk, and conversely, which ones are at the lowest risk as well? What are the important mechanisms that influence the species response to change? And does provenance selection ameliorate the risk to climate change for some of these species? So this has, would really like to think, some really key benefits for Melbourne water. One, I mean, understanding what are the most resilient species is comforting when you plan forward. So if you put out, vegetation in the field today, tomorrow, next week, next year, next 10 years, being confident that it will last a long time. We're also really thinking it's important that trying to pull apart what the role that provenance, climate of origin of seed supply, and genetics means for species resilience. So it might be that the current genotypes or phenotypes we're using might be quite sensitive or vulnerable to climate change. However, potentially there are populations within that species that we could use. So we don't have to change the species. We can just change the genetics. We also want to identify key mechanisms. So one of the things that we do a lot of work on is understanding the role of regeneration, so germination, for example, in those key processes. And this has implications because some species can be quite sensitive in their germination phase, but not so much in their growth phase. So that means you can overcome some issues of sensitivity to climate change, for example, by planting versus putting out seeds. Of course, this research hopefully will identify some priority gaps for further research. This is a pilot study, after all, and we really want to be able to, we're not going to be able to address all the unknowns. We really want to, test some of these ideas out and see if there are identify these priority gaps. And then overall, all of these together will help us develop climate smart revegetation practices. So really be able to put in place a plan and knowledge that will help adaptation to climate change. Methodology. So we've got a fairly complex study design involving both modeling and laboratory work and experimental plant experiments in glasshouses. So there's two types of modeling going on. We're using species distribution modeling, which takes presence-only records of species, and we generate some pseudo-absences points, and we model the response of these points to climate and come up with models to predict their climatic niche, both today and in the future. We're also undertaking seed ecology experiments to parameterize a process-based model called TACAGEM, which is a model that models the ability of plants to regenerate through germination and then the ability of those germinants to survive over time and grow into seedlings and saplings. And so to do that, we have to do seed ecology experiments, looking at growing degree days, how fast they regenerate or germinate, sorry, look at drought tolerance within germination, and also we're doing a drought to look at their ability to withstand drought after they germinate. And we're doing this modeling over here with 31 species, but we're doing this with 6 species over here. And also with one of these species, we're really diving into looking at the role of genetic provenances. So those 6 species are lightwood, the Agonia siberianae, Alocasarina, Bracharia, Alaria lyrata, and then river red gum. And with the river red gum, this is the one where we're doing quite a bit of genetic work with to understand the role of genetic variation. So to date, we've done an extensive literature review on the seed ecology of those 31 species. We've built species distribution models for those species. And for a subset of these, we've actually developed 26 additional models because we had enough information about subspecies. So what we're really interested is using that as a proxy for genetic information And like, okay, do we get similar responses through the subspecies analysis? And we can identify particular subspecies that might be actually better for Melbourne's future climate. So we've created map predictions for 57 species slash subspecies under both current and future climates. We've sourced seeds for those 6 species that I mentioned earlier, and that's from one province of each for the seed trials that we've done, and those are all completed now. We're doing the mechanistic modeling of those 6 species to a changing climate using TACA-GEM. We've got seeds from 14 provinces of river red gum, and we're undertaking the seed trial of that in progress, and then we're growing these on to undertake drought trials in the near future. So just a little example of Eucalyptus canadensis. This is all subspecies. So if we take every single record of Eucalyptus canadensis that's available, put them all together, and predict its climate, what we can see is, in the Melbourne area here, there's fairly low probability up in this, but high probability there. And as we go to one of the potential future climate scenarios, we can actually see that Melbourne becomes a bit more suitable for Eucalyptus canadensis. But again, this is all subspecies, everybody combined. So we've got different models and different responses for the different subspecies that are out there. So what do we propose for next year? We're proposing to do some drought trials for these 6 species. So that will help with improving the TAC and GEM model in particular, because we can then look at longer term drought responses. So when things go from germinants and grow into seedlings and saplings, we're going to conduct these drought trials with those 14 river red gum provinces as well. So we'll be able to really test again in greater detail that role of genetic variability in resilience. We're undertaking the genetic testing of those seedlings as well, and that will be a really important element because we want to understand how much is genetic and how much is phenotypic, and we understand what genes are being expressed in that drought response. That's really important because it means when we start to look for drought tolerant populations out in the field, we'll be able to look for those particular genes. So that will speed up the process of selecting future genotypes that are more adapted to Melbourne's future climate. And we're also wanting to establish a province trial with these river red gums across a natural rainfall gradient. So setting up trials from the west to the east of Melbourne from low to high rainfall. We want to be able to actually put these provinces in the ground and monitor them long term. All this work to date has been in growth chambers and in glass houses. We want to put these in the ground and look at them longer term. And this is important because there's this thing called cryptic maladaptation, meaning that we could have great responses from certain provinces and then one bad winter, one bad summer. So an extreme event mistimed can lead to high rates of mortality and failure. And we want to know that um, before we implement kind of broader scale planting of different genotypes and phenotypes in the region. And this would hold true for many species. And we're just using the river red gum as a starting point. I'd like to acknowledge Melbourne Water. They've been incredibly patient with us, particularly given the impacts of COVID that's has had on us and our ability to do this work. The University of Melbourne team, I'd like to thank Jung and Satcha, Linda, Andrew, and Stefan Arndt for their input and advice. Linda, in particular, has been absolutely amazing driving this project. She even took the climate growth cabinets home, and we had them in her laundry room during COVID to get some of this work done. Adam Miller from Deakin University has been super helpful helping us get our genetic stuff up and running. And I also think Greening Australia been a big driver to getting this project up and running. A big shout out to Satcha when he was at Greening Australia and now Melinda. So if you have any further information, please feel free to give me an email and more than happy to take any questions. So thank you very much.